but I'm really happy to have him because especially he's the world-renowned expert in T-cell development. So I think you probably should share my enthusiasm and to have him as a T-cell lecturer in our class. Okay.
Thanks, Jimmy. The memo stick is.
Oh, I'm sorry.
Pick it up.
Yeah.
Left
Yeah.
the door.
Yeah.
Charlie.
Okay.
Pointer. Okay. So I'm really going to concentrate in this talk on how T cells become activated by antigen expressed on antigen-presenting cells and the decision-making that's involved in whether or not you generate a normal effective immune response or tolerance. And this is really some of what I'm going to talk about. You may have heard to some extent in previous lectures, and some will be new to you. So I'm going to begin by setting the stage for how T cells interact with APCs by talking about, very briefly, about T cell development, how an animal generates a mature repertoire of T cells. And so I'm going to just briefly touch on how T cells arise in the thymus. So for me to advance this, I click on the left.
So probably you have to use the keyboard.
Oh, this is okay.
Oh,
Yeah.
okay.
Okay, so as you probably know, most T cells present in the thymus are immature and they express both CD4 and CD8 cells, so we call these double positive cells. And these cells arise from stem cells, which represent only a small proportion of thymocytes. And I'm not going to say anything about how these so-called double negative cells differentiate into double positive cells. I'm going to emphasize it is these cells in the thymus that express this intermediate density of the alpha-beta T cell receptor, which of course is expressed at a high level on mature T cells in the extra-thymic environment. And these cells, by a process of what we call thymic selection, give rise to these mature T cells, CD4 single positives and CD8 single positive cells. Now, if you think about the thymus, the first thing to emphasize is that it is naturally a very large organ. So in young infants and young baby mice, the heart is actually as big as, or even bigger than the heart. Now, I emphasize that point because this, strangely enough, was realized relatively recently, especially by physicians. So, back in the 1950s, most physicians who are not especially well trained, I'm a physician, when they thought of the thymus, they thought of autopsies. And autopsies are generally conducted on people who die from some chronic disease. And chronic disease induces stress, and stress, which the thymus is extremely sensitive to, causes the thymus to shrink. So most physicians think of the thymus as being, or did think of the thymus as being a very small atrophic organ. And it really only became appreciated as being a large organ like this to physicians when chest x-rays became available as a clinical tool. And that was around about the 1950s. And so what used to happen, not infrequently, is a child would be brought in by its mother to the doctor, and the child, as many kids do, had a chronic cough, and they were wheezing and spluttering, etc. And the physician would sound the child's chest and saying, oh yes, you've got an infection there. But then he says, well, we have this new tool called a chest x-ray, so let's have a look at the chest. So the physician would take an x-ray of this young child's chest and see, to his surprise, that the thymus was very big, because he normally thought that the thymus was quite small. And so he noticed that the thymus was actually wrapped around the child's trachea, which of course the normal thymus is, and he reached the conclusion that the reason that this child was coughing and wheezing is because of chronic strangulation by the thymus. So the treatment that was advocated for this disease of status thymolymphaticus, as it was called, which is basically just a normal thymus, was to irradiate the thymus of children. And this was actually done routinely by physicians in the 1950s. Extraordinary, really, if you look back on it. And now, of course, we know that this was a really stupid diagnosis, and the thymus is normally very large. And then it shrinks at around about the time of puberty. It continues to function into old age, but most T cells are generated in young life. And after the age of puberty, you really don't need the thymus because T cells are actually very long-lived. Okay, so the thymus, two zones, the cortex and the medulla. So this is the cortex where most double positive cells sit, and they are wrapped around this dense network of cortical epithelial cells, which are expressing MHC molecules, which is crucial for thymic selection. And in the cortex, there are very few bone marrow-derived cells. The bone marrow-derived cell sits largely here in the medulla. Now, this is actually a picture of a human thymus that have these characteristic Hassel's corpuscles, and I'm not sure that anybody quite knows what these things do, but histologically they're quite dramatic. You don't see them in the mouse, which is what I work on. But they are the type of epithelial cells. Now, the bone marrow-derived cells are largely dendritic cells and a few macrophages. And these cells are very important for presenting antigen to T cells. Now, I should mention that in the cortex there are very few dendritic cells, but there are macrophages, and their duty, as I'll come to in a moment, is to eat dying thymocytes. Now, in terms of self-tolerance induction, where you remove cells with overt self-reactivity, it's important to mention that most of the self-peptides expressed on the MHC molecules in the cortex are ubiquitous self-peptides. In other words, peptides that are synthesized by every cell in the body, which are basically the breakdown products from normal proteins. But in addition, self-antigens such as, say, insulin in the beta cells of the pancreas, they can be carried into the thymus by dendritic cells in small amounts. That's one point. And secondly, there's a lot of interest in the expression of so-called AIRE genes, A-I-R-E genes, which are transcription factors that lead to the selective expression of a variety of self-antigens like insulin or like thyroglobulin by the medullary cells in these medullary epithelial cells here. And each of these medullary cells can express a variety of these self-antigens, antigens that you normally think are expressed selectively in the thyroid or in the pancreas. They're actually expressed in low levels in the medulla of the thymus. And cells with strong reactivity to these proteins are actually deleted. And that's a very important mechanism of self-tolerance. And in fact, in mice and people who lack the AIRE gene, then this deletion process cannot occur and you will develop autoimmune disease. Does that make sense? Okay. All right, so let's think about how cells are selected in the thymus. This is the so-called affinity model of thymic selection. In other words, the fate of T cells generated in the thymus depends on their relative affinity for the peptides expressed on the MHC molecules expressed in the thymus. Now, the first important point to emphasize is that of the double positive cells present in the thymus, the vast majority of these cells, and I've written down here, around about 97% or so, die in situ in the thymus. They are of no use. So when you think of thymic selection, it's an extraordinarily wasteful process. Only about 2% of cells are actually selected for export. And these cells, the 97% or so which die, they die because they get no signal through the TCR. So because these cells receive no signal, they die from neglect, as we call it, no signal. Now, positive selection. This is when a small proportion of double positive cells, around about 2% or so, has some low but significant affinity for the ubiquitous self-peptides expressed on the MHC molecules on cortical epithelium. Not too high, because that's going to kill them as we come down here, and not too low. If it's too low, they will die by neglect. Just low but significant. And that will give a weak signal through the T cell receptor. And that weak signal basically tells the cell, don't die, survive. And this avoids death by neglect. And these cells are then selected for export from the cortex into the medulla. Now, those cells, most of those cells will then make their way out of the thymus. But of the cells that undergo positive selection, some cells will actually have relatively high binding affinity for the MHC molecule. So if those cells were allowed to get out of the thymus, then they would pose the risk of responding in a vigorous fashion and inducing autoimmune disease. So these cells have to be destroyed. And they're destroyed because they have strong reactivity, because TCR signaling is strong, and they cannot, these immature cells, unlike mature cells, they cannot withstand strong signaling, and so they die. And this is called negative selection. Now, as I mentioned, it involves cells that happen to have high affinity for ubiquitous self-peptides, peptides that are expressed on all cells in the body, and also cells that are reactive to these self-antigens like insulin that are brought into the thymus by APCs or actually synthesized by medullary epithelial cells. So all cells that have high reactivity for whatever self-antigens are expressed in the thymus are killed because of strong signaling. So this is the so-called affinity model of thymic selection. Now, I should mention that there are some cells that have intermediate affinity between the affinity required for positive selection and negative selection, in between. And these cells actually have sort of relatively strong affinity for self, but not strong enough to actually kill the cells. Now these particular cells, at the level of CD4 cells, are the cells that become so-called T regulatory cells, which I'm sure you've heard about in great detail. As you know, the T regulatory cells play an important role in limiting the reactivity of typical mature T cells. Okay, now negative selection and lack of positive selection. The notion that most T cells actually die from neglect in the thymus had been around for a long time, but the problem is people couldn't really see any evidence of cell death. You look at a histology of a normal thymus and all you see is live cells. So this idea that there's massive death in the thymus seemed, well, there was really no evidence for it until about 15 years or so, people developed methods, histological methods, for defining cells undergoing apoptosis. And this method is called tunnel, and it's a way of basically picking up dead cells in sections. And Charlie Sear, who was in my lab at that time, used this method to look at cell death in the thymus. And this is these are some of his pictures here. This is a normal thymus here. This is the cortex, and this is the medulla here. And you see lots of orange cells here. Now, what are these cells here? They're macrophages, and they're packed full of dead dying double positive cells, cells that have failed positive selection. So individual macrophages will eat lots and lots of these dying BP cells. And that's where these dying cells go. They get eaten by macrophages. Now, if there is a large presence of self-antigens in the medulla in artificial systems, then the cells that have high reactivity for these self-antigens, when they move from the cortex to the medulla, then they receive a strong signal and that will kill them. And this is one of Charlie Sir's pictures here of massive numbers of autoreactive T cells reacting to a particular self-antigen in the medulla, and these cells are being eaten by antigen-presenting cells in the medulla itself. So that's all I want to say about negative selection. Point to recapitulate is most cells die in the thymus. The question is, why has evolution come up with this apparently very wasteful procedure where 98% of cells or so die there? Well, the arguments about this have been going on for the last 30 years, and people still argue, in fact. But I think most people go along with the idea now that it's all about the fact that mature T cells, when they reach the secondary lymphoid organs, they don't live by default. They have to be signaled to live. And the question is, what keeps them alive? Now, you might say, well, what's the evidence that T cells actually have to be signaled to survive? And for any of you that work in mice, you know that if you take normal T cells from the spleen or the lymph nodes, and you put those cells in tissue culture without any stimulus, the next day they're all dead, or most of them. You can keep them alive if you add certain growth factors or antigen-presenting cells, but you have to add something to keep them alive. So what keeps the cells alive under in vivo conditions? Well, we now think that what happens is that when the cells are selected in the thymus, they're selected to have some reactivity to the self-ligands, the ubiquitous self-peptides linked to MHC molecules in the secondary lymphoid organs. And so, in that sense, respect, we know that when naive T cells leave the thymus, to keep them alive, they have to have continuous contact with self-MHC ligands in the spleen and the lymph nodes. And this so-called TCR tickling gives them a low level which says don't die, and it leads to the upregulation of molecules which normally keep the cells alive, like BCL2 and so on. Okay? When these cells eventually see antigen and differentiate into memory cells, then, as Charlie Sir will talk to you next week, excuse me, these cells don't need to see self-MHC. They're kept alive by cytokines. Okay, now naive T cells, which is what I'm talking about now, they have to be kept alive through this interaction with self-MHC ligands and also a cytokine IL-7. Now, this interaction does not make the cells divide. It keeps them alive in interphase. And that's an important point to bear in mind. So here, let me briefly talk about the lifespan and migration of normal T cells. Now, normal T cells, when they come out of the thymus, they go round and round the body, basically looking for antigen. That's what their duty is. So you can view them as a sort of army of soldiers looking for the enemy. And they do that, as I'm sure you've heard in previous lectures, through defined areas of the lymphoid tissues, the spleen and the lymph nodes. Let me just touch on a couple of points here. So they do that by migrating through the lymphoid tissues from the bloodstream and then into the lymphatic organs, of which the thoracic duct, which is a lymphatic vessel here, is the chief vessel. And they do this naturally. And in mice, you can stick a cannula in the thoracic duct of mice, which is something that I spent many years doing when I was a postdoc and after that. And you can collect enormous numbers of lymphocytes from a thoracic duct cannula. So a normal mouse has in the order of 2 by 10 to the 8 lymphocytes. And if you stick a cannula in the thoracic duct of a mouse, overnight you can collect half that number of cells, something like 10 to the 8 cells. And these are very useful and totally alive cells that you can do lots of nice experiments with. The trouble is now that the animal care people take great exception to people doing this operation, especially in the United States, and it's extremely difficult to get this past the committees. So I've more or less given up doing that now. But the point to emphasize is that by looking at cells in thoracic duct lymph, this was the classic way that people like Jim Gowans 50 years ago demonstrated lymphocyte recirculation. Now, what do we mean by lymphocyte recirculation? We mean that when cells are in the bloodstream, they move through the lymphoid tissues, especially lymph nodes, go through them, and then exit through into the efferent lymph, and I'll come to that in a moment, and then enter the thoracic duct. And so you can take radio-labeled lymphocytes, T cells, inject them intravenously, and then you watch the appearance of these cells in thoracic duct lymph, and they will appear after 12 to 24 hours or so. And that is blood to lymph recirculation. So let's just briefly think about how lymphocyte recirculation happens. It happens when the small blood vessels percolate through lymph nodes, and the tributaries of the small blood vessels are called high endothelial venules. I'm sure you've heard about these before. And normal resting T cells have so-called homing receptors like L-selectin, and they make contact with ligands. Here there's the sulfated glycam molecules here on the endothelium, and that allows them to stick and with other molecules to make their way into the so-called T cell zone, which is this area here in light blue. And this is the B cell zone here. And then these cells will wander around in this paracortical region for a few hours, and then they make their way down to the medullary region of the lymph node, and go into the medullary sinus, and then into the efferent lymphatic vessel, which then drains into the thoracic duct. Now, this is the afferent lymph, and there are very few lymphocytes in afferent lymph, very few small resting T cells. There are some activated memory cells, but no small T cells, or very few. The point to emphasize, and this is often forgotten, is that lymphocytes get into lymph nodes not through afferent lymph, but through these high endothelial venules. So this entry to high endothelial venules is controlled by these homing molecules, and I mentioned L-selectin that binds to these so-called vascular addressins. And then there's another very important homing receptor called CCR7, and that's a chemokine receptor, and that binds to chemokines, CCL19 and 21, which is synthesized by various cells just inside the lymph node, close to the high endothelial venule. And that allows the cells to get into lymph nodes, and then they use different methods to move through the lymph node into the efferent lymph. Now, when T cells actually, as they're wandering by recirculation around the body, they actually do see a foreign antigen, then the cells that have reactivity to this antigen are trapped. And that means that if you're collecting the cells in the lymph, this is looking at numbers of antigen-specific cells in the lymph, for a period of a couple of days or so, those antigen-reactive cells are gone from the lymph. And that's because they've all accumulated in a particular lymph node. In other words, a lymph node where the antigen is expressed. And then, after the immune response is generated, these effector cells then move back into the lymph, and the lymph drains into the bloodstream, and the bloodstream carries these effector cells randomly throughout the body and they will make their way back randomly to wherever the pathogen is situated, in the lung, for example, with influenza infection. And then they will attack the virus-infected cells in that organ. Okay, so just very briefly, let's just think of how an immune response gets initiated. So in the case of whole bacteria, they can come in through the afferent lymph, they could get picked up by macrophages, and they're largely destroyed there. But some of the antigens get picked up by immature dendritic cells, and these then enter through the afferent lymph, they enter the paracortex, where they present their peptides bound to surface MHC molecules to T cells, CD4 cells and CD8 cells. And then these cells will mount an immune response, divide, differentiate into effector cells, move into the medullary sinus, into the efferent lymph, and then they're carried throughout the bloodstream. Okay, so now let's think about T cells interacting with APCs at a more intimate level. Now, ultimately, and you've heard lectures on antigen presentation before, you know that when APCs ingest antigen, then ultimately what happens is that peptides derived from the pathogen are presented on the surface of MHC molecules. And then reactive T cells see these antigens, and if the dendritic cell is mature, then they will start to respond. Now I want to talk about the requirement for co-stimulation in some detail. So let's think of how the response gets started. The first thing is when lymphocytes move through a lymph node, which they do the whole time in the absence of antigen, they're constantly screening the existing dendritic cells in the T cell zones for foreign antigen. And they do that by making short-term, transient contacts with APCs. And they do that by using molecules like LFA1, which is an adhesion molecule, and it has intermediate binding affinity for its ligand, ICAM1, on an APC. And so that will allow short-term contacts, and it will allow them, the T cells and the APCs, to come together. And that will then give the T cell receptor on that cell the opportunity to see if the MHC molecules on the APC are expressing a foreign peptide, which is shown here in red. Now, if they do, then the TCR will then bind to the MHC peptide shown here. And then you have this signaling cascade, which leads, amongst other things, to so-called inside-out signaling of this LFA1 molecule. And that will change the three-dimensional structure of the molecule such that its affinity for ICAM1 increases. And now it has high binding affinity for ICAM1. So that means that when the T cell receptor is bound to this peptide linked to MHC, and then you've got the signaling here, and now this high affinity LFA1 ICAM interaction stabilizes that interaction, and in conjunction with a whole variety of other co-stimulatory molecules, you have an effective synapse formation. So, broadly speaking, under these conditions, you have this interaction between the T cell receptor and MHC and peptide, strengthened by LFA1 ICAM interaction, and also strengthened by the interaction of molecules like CD4 and CD8 on T cells. Now, you've probably heard that these molecules actually function by having binding affinity for monomorphic epitopes on class 1 and class 2 molecules. So CD4 will bind to a monomorphic epitope on the class 2 molecule, and that provides adhesive function and basically strengthens this interaction between the T cell receptor and MHC. And for CD8, it's the opposite. The CD8 molecule binds to epitopes on the class 1 molecule. Now, generally, it's thought that this is so-called signal 1. It gives a signal to the T cell, which, as I mentioned before, signals the cell, leads to inside-out signaling of LFA1 and so on. But that's really not enough to mount an immune response. In addition, you have to have what's so-called signal 2. So let's think about signal 2. And here, the most important molecule initiating signal 2 is CD28. And CD28 binds to ligands, and there are 2 of them, B71, B72, also called CD80 and CD86. But let's just call them B7 on the APC. And this provides, or is thought to provide, a separate signal called signal 2, which is crucial for an effective immune response. And this is all going on at the level of a synapse, which is shown diagrammatically here. And the synapse, which is basically the interface between the reactive T cell and the APC, it has a whole variety of molecular interactions going on. And the T cell receptor is right in the middle of this synapse. And so the T cell receptor is here, it's recognizing peptide and MHC. And in addition, the costimulatory molecules CD28 and B7 are also in this inner region. So that's shown here and here. And these 2 sets of interactions collectively lead to intracellular signaling, which involves activation of CD3 and these molecules like ZAP70, LCK, PI3 kinase, and so on, which leads the cell to proliferation and differentiation. Now, there are also these molecules that have the opposite function, like CTLA4, which is an inhibitory molecule. And there's a lot of interest in how CTLA4 inhibits. And I don't think this really has been settled yet, but one of the things that CTLA4 does do is it binds with very high affinity, higher than CD28, to these B7 molecules. And so CTLA4 basically competes for binding to B7, thereby depriving CD28 of binding to B7. In fact, CTLA4, I think, has about 100-fold higher binding activity for B7 than CD28 does. Now, in it, so that's one function. It just competes for B7, and also it's linked to these inhibitory molecules that shift to intracellularly. Now, so there are a whole variety of these molecules in the inner portion of this synapse, which I won't go into. But also in the outer portion, this is where LFA1 ICAM interactions, this adhesive function, this is where they are situated, and they more or less amplify the activity of these molecules in the center. And then you have other molecules like CD45 and CD43, which are even further outside this synapse. Now let's think a little bit more about this so-called signal 1, signal 2 idea. So as I mentioned, TCR binding to MHC peptides thought to be signal 1, CD28 binding to B7 is signal 2. Now CD28 B7 interaction has been around for at least 20 years now. But exactly what it does is still debated. And if you read articles about it, it's all very confusing. Nobody seems to be able to quite decide what it does. I think one of the things it does very well is to act as an adhesion molecule. And you will not see that mentioned very much. But in fact, CD28 B7 interaction is quite an efficient adhesive interaction. But anyway, the paradigm, the idea that has grown up in immunology over the last 30 years or so is that you have to have these 2 signals to get immunity, and if you only have signal 1 alone, then you have tolerance. So what does this actually mean? Well, let's think of where this idea actually came from. And it actually came originally from studies on B cells, published about 30 years ago by Mel Cohn, who's a well-known aged immunologist now at the Salk Institute in San Diego. And these guys basically said that in order for a B cell to be activated when it sees an antigen, just recognition of antigen is not enough. There had to be something else that they call signal 2. And we now think of that largely as being the provision of T cell help from CD4 cells. And then this idea for B cells was adapted to T cells a few years later by these people here. And there it was argued that you have to have a second signal, that recognition of MHC and peptide by the TCR was not enough. And then the crucial experiment that was conducted at the NIH about 20 years ago by Mark Jenkins and Ron Schwartz was to show that if you cultured T cells with normal APCs presenting antigen, well then you would generate an immune response in tissue culture. But if the APCs were fixed by chemical fixation by this compound ECDI, then these fixed APCs would not generate an efficient immune response. And instead, the T cells were rendered anergic, in other words, unresponsive when they were subsequently exposed to antigen, same antigen presented by normal unfixed APCs. And so this was regarded as really the first direct evidence that signal 1 alone is tolerogenic. And so the argument here is that chemical fixation prevents the delivery of signal 2. So then, of course, the issue has always been, well, what does signal 1 alone do? Why is it tolerogenic? So what we think is probably going on here, I mean, the question is, well, why does chemical fixation prevent the delivery of signal 2? And here it's important to bear in mind that, as I'm sure you've heard in previous lectures, the expression of molecules like B7 on normal APCs, on immature dendritic cells, is low. And so it's not sufficiently effective to generate a good immune response. And what happens during a normal immune response is the B7 is upregulated to a high level. So you get a high expression of signal 2. And B7 is normally upregulated if you have an infection by the TLR agonists expressed like LPS by bacteria and various other stimuli. But in the case of this chemical fixation experiment of Jenkins and Schwartz, the point to bear in mind is that if you take normal APCs, immature dendritic cells, and you put them in tissue culture, then these APCs think the plastic is a foreign substance. And they try to ingest the plastic, which of course is impossible. But that attempt at ingesting the plastic actually activates the APCs. Now that's probably what normally goes on in vivo when an APC tries to ingest something really big like a sheep erythrocyte, which is a very immunogenic structure. You don't need adjuvants to get a response to sheep red blood cells, just because it's big and it can activate APCs. So when you put normal APCs in tissue culture on plastic, it activates the APCs. Whereas normally you have to have so-called adjuvants, which are TLR agonists, to activate the APC. So in these chemical fixation experiments, what the people did is that they put the APCs in culture and then immediately fixed them And that prevented the cells from being activated by the plastic. And that's why signal 2 was not delivered. And in fact, it was subsequently found that if you allow the APCs to sit in plastic and then you chemically fix them, then they're immunogenic. So this then still asks the question, what does signal 1 alone do to a T cell? If the cell just sees MHC plus peptide alone, what does it do? Does it really make the cell immunogenic? So what's the evidence for this? Well, the usual explanation for this is that if you take naive T cells in tissue culture and then you expose them to an artificial TCR signal, cross-linked anti-CD3 antibody, this will make the cells divide a bit, but it's not a very efficient signal. If you want a really efficient signal in culture, you have a combination of anti-CD3 and anti-CD28, and that's a very good stimulus for T cells. But anti-CD3 alone doesn't produce much IL-2, which you generally need to expand the cells. And so the cells divide a little bit, and then in the absence of IL-2, then they just die. But if you think about it, it's not a very physiological provision of signal 1. Because here you're cross-linking CD3 on the surface of the T cell, and that's not what occurs naturally if a T cell sees MHC and peptide alone without co-stimulation. This was of some interest to us some years ago, so I just want to briefly talk about how we approach this problem. So optimally, if you really want to look at the consequences of a cell encountering just signal 1 alone, you want to have an APC that expresses just MHC and peptide and nothing else. Now you can do that up to a point by using APCs that lack B7. But the problem is that those cell lines, even though they lack B7, they'll express a whole variety of other molecules which can provide some form of co-stimulation. And so under those conditions, in the absence of B7, you will see roughly what you see with when you expose cells to anti-CD3 alone. They will proliferate a little bit and then they'll die. But the problem is that these cells are still providing weak stimulation by molecules other than B7. So what you really want is a cell that only expresses MHC and peptide and nothing else. And you can't do that with mammalian cells, because every cell that you look at is going to express some molecules that provide some adhesive function for complementary molecules on the T cell. So we approach this by taking the argument that if you move far enough away from mammalian cells in terms of phylogeny to insect cells, there would be nothing on the surface of an insect cell that could be seen by any molecules on a T cell. And so what we did is we took insect cells, Drosophila cell lines, and we transfected them with mammalian molecules and asked the question, what do you need to transfect into an insect cell to turn it into an efficient APC? And so we put in MHC molecules, B7, ICAM, and so on. And the rationale then is there's nothing on the surface of a Drosophila cell that can be seen by T cells, and all our subsequent work has confirmed that as being true. Although Drosophila cell DNA, because it has CpG motifs, can stimulate. So you have to make sure you don't have DNA in the system. But I won't talk about that. Okay, so what do you see with this system? Well, if it turns out that if these insect cells express just MHC and peptide alone, and here we use a TCR transgenic system, and you can use either CD8 cells, as shown here, or CD4 cells. It works in both situations. just MHC and peptide alone, even in high concentrations, you ask the question, do you see signs of T cell activation? Do you see proliferation? R2 production? The answer is no. You see nothing. The only thing you see is internalization of the T cell receptor, but not activation of the T cell itself. And so the conclusion from these experiments is that signal 1 alone, paradoxically, does nothing. But you can tell it does not lead to initial proliferation followed by energy induction. Whereas if you put in MHC peptide together with B7, you will see an efficient immune response. The cells will proliferate and produce R2. If you put in ICAM, they will proliferate a bit, but very little IL-2. But if you put a combination of both B7 and ICAM, then you see a very efficient immune response. Now, I may mention that this, of course, is a highly artificial approach, but you can use this approach to ask questions like, what does signal 1 alone do? But you can also use it for other things. And when I was doing this work at Scripps with these insect cells with a colleague of mine, he moved from Scripps to Johnson & Johnson, which was just across the road from Scripps. And he immediately decided that what works in the mouse could work in humans. And so he decided that he was going to cure cancer using these fly cells, which sounds a bit far-fetched. But what he and his collaborators did was to make a panel of insect cells transfected with human MHC molecules, HLA-A2, human B7, ICAM, and other molecules that they've since put in. And then if you're presented with a patient with cancer, melanoma, then they would take CD8 cells from the patient by leukapheresis and collect relatively large numbers of these CD8 cells, and then culture them together with these insect cells expressing these human molecules together with melanoma peptides. And they do this routinely now at Johnson & Johnson. And they grow up these CD8 cells stimulated with the melanoma peptides from these cancer patients. The cells expand, you add cytokines, you expand even more. These cells can be fixed by UV light, and so the FDA does not regard them as being insect cells anymore because they're basically killed. And then you can expand them, and then you can inject them back into people, and you can inject 10 to the 9 cells quite easily, of which maybe 50% of these cells are actually antigen specific. And there are clinical trials that have been going on now for several years. None of this has been published, and in some patients you actually do see remarkable regression. So this is one situation where fooling around with insect cells and mice can actually lead to something that may ultimately be useful clinically. Okay, but anyway, this is a sidetrack. The important thing the message I want to get across is that this experiment says that as long as you're dealing with cells that express no co-stimulatory molecules, then just signal 1 alone actually does extraordinarily little.
Sean, can I ask a question?
Sure.
So if you compare the T cell without an insect cell versus, or the insect cell with no MHC molecule versus just MHC peptide,
Yeah.
how about the survival rate? Or the
Survival rate of the...
T cells? Does it change the half-life of T cells at all? Or not?
You mean in culture? Or?
Yeah, in culture.
Well, the T cell, you're talking about signal 1 alone, or?
Signal 1 alone versus no signal at all.
The answer is they both die at the same rate. So you don't protect them at all. As I said, the only thing you can see is internalization of the TCR, but no apparent upregulation of BCL2, and they just die.
There must be some kind of signal delivered to
There must be some sort of signal.
right? I mean, if the
Right.
question would be...
But that signal is presumably so transient in the absence of signal 2 that it just basically doesn't do anything that you can see. Okay, so the message here then, paradoxically, is that signal 1 alone does extraordinarily little, and signal 2 alone itself really doesn't do anything. I mentioned before that generally ligation of CD28 doesn't do anything. And there's this long-term debate that I mentioned about what does signal 2 actually do, and people still can't decide on this. But I will, a little later, present you this extraordinary evidence from one particular antibody to CD28 called, with this complicated name here, that had very, very dramatic effects. In other words, it does give a signal. I'll come to that in a moment. Okay, so optimally, the signals needed for T-cell activation. So signal 1 is TCR interaction with MHC and peptide, and by itself that doesn't seem to do very much that you can detect. And then signal 2 is CD28 interaction with B7. But there are a whole series of other molecules that provide co-stimulation or adhesive function. Quite what these molecules do is still a little bit unclear. So I mentioned LFA1 ICAM interacting with ICAM1. There's another molecule 4-1BB that interacts with a ligand, the CD27, interacting with CD70, OX40, and so on. And all of these together strengthen the interaction between the T cell and the APC and intensify the overall signal and lead to efficient T cell activation. Now I should mention that in order to get an efficient immune response, you have to have cytokines. And the cytokines are crucial for expanding responding T cells. And for CD8 cells, you need IL-2 for the expansion, but to keep the cells alive and proliferating, certain other cytokines, particularly in type 1 interferon and IL-12, of great significance. So if you, in mice, generate an immune response and the T cells are taken from a mouse that lacks the receptors for type 1 interferon, you will get a very poor response. And the same thing depending on the infectious agent if the cells can't see IL-12, even though IL-2, etc., is being produced. And this is sometimes called so-called signal 3. And please interrupt and ask me questions if you have other things you're uncertain about. So, operationally then, you have to have these 3 signals to get an efficient immune response. And if you don't, then the argument is that you can have some form of tolerance. And the typical situation here is that when T cells see MHC and peptide on an APC that has not been activated by an adjuvant or by TLR agonists, in other words, where the density of B7 and other co-stimulatory ligands is low, then you have tolerance. And so under these conditions, the cells will proliferate, they will go through a refractory stage called anergy, and ultimately a lot of the cells will be deleted. And so the current view then is that when you do see tolerance developing in an immune response, this is not just signal 1 alone that the cells are seeing, it's signal 1 alone together with a weak signal 2. And that happens particularly if the APC has not been efficiently activated. And this probably is an important mechanism for why we normally do not develop autoimmune disease. Autoimmune disease is, for the most part, a relatively rare phenomenon. So some people will develop type 1 diabetes, but most people don't. So why is type 1 diabetes and other forms of autoimmune disease relatively rare? Well, I mentioned that many T cells are reactive to insulin. In the case of type 1 diabetes, are actually deleted in the thymus because APCs carry insulin into the thymus and because some of the medullary epithelial cells express insulin and lead to deletion. But in addition, you have the production of energy of cells with reactivity to, say, these pancreatic beta cell antigens, beta cells being, as you know, the cells that produce insulin. So let me just show you this nice experiment conducted a few years ago now by this group. And what they showed is that when beta cells are dying in the thymus, and in certain strains of mice, you can induce the beta cells to die slowly. Then, and this may be well be occurring naturally in patients that develop type 1 diabetes. Normally, these cells are internalized by immature dendritic cells. So they're immature in a resting animal or a mouse. The dendritic cells in the draining lymph node of the pancreas are resting. There's no adjuvant around, so they're resting. And so they will be, these antigens will then be loaded onto the surface of the APCs, and in the case of class 1, the MHC molecules will pick up insulin peptides through what's called cross-presentation, which I'm sure you've heard about. And then recirculating T cells will come across these self-antigens on these dendritic cells in the draining lymph node, and they will mount an immune response. But that response will be abortive because the dendritic cells do not express a high density of B7, they're immature, and so it's abortive, the cells will become anergic, and the cells will die. And this is shown diagrammatically here. So here's a pancreas, and here are islet beta cells. And so that these cells, if they die, then their internal proteins can be taken up by, they move to the draining lymph node, taken up by dendritic cells, and they can be then go through the class 2 and the class 1 pathway, and they'll be loaded onto the surface of MHC molecules, and on these immature DCs, they will be seen by these cells here, these CD8 cells, and because of the lack of good signal 2, then these cells will die. So this is probably an important mechanism for avoiding self-tolerance. So I mentioned that in the thymus, a lot of T cells with reactivity to antigens like insulin are destroyed by negative selection. And then outside the thymus, this constitutive release of self-antigens from organs like the pancreas, as long as these self-antigens are taken up by resting DCs, not activated DCs, then there will be an immune response, but it's abortive, and you'll have anergy and eventual death of these cells. So the danger, of course, is that if this process happens and you have to have adjuvants around, which can be provided by an infectious agent, and then the response can be much more efficient, and then there's the danger of autoimmune disease. And it's under these conditions where you need yet another backup mechanism for tolerance induction, and that is the influence of T regulatory cells. Now, you've probably heard a lot about T regulatory cells in previous talks, and I mentioned that these cells are naturally produced in the thymus. As part of thymic selection, they're thought to have intermediate affinity for cell proteins, and they come out of the thymus, and they are also reactive to various cell components, and they're relatively long-lived. And when they see some self-antigen like beta cell antigens in the thymus, in the draining lymph nodes of the pancreas, then they can respond, and they will reduce the function of normal T cells. And in addition to these so-called naturally occurring Tregs, it's become apparent in the past few years that you can actually generate Tregs from normal T cells. Now, a Treg, by definition now, is a cell that expresses this transcription factor called FOXP3, which I'm sure you've heard about. And that basically is a negative regulator, so it prevents the responding T cell from producing IL-2, which is crucial for the immune response. And one of the main functions of Tregs, but there are many functions, people debate what these Tregs actually do. How do they suppress the function? Well, one of the things they do is that they will express receptors for cytokines like IL-2, but they don't produce IL-2 itself, and so they will absorb IL-2 from other cells, act as a sort of sink. And they consume cytokines. They do lots of other things in terms of their suppression. So it's become apparent that these cells can be generated from normal T cells during normal immune responses. And what are the requirements for inducing a normal T cell responding to a normal antigen to become a Treg? In other words, a cell that's expressing FOXP3 and has suppressive function. A lot of debate about this. But one thing is clear, that if you have the presence of certain inhibitory cytokines like TGF-beta, this is crucial. But the rules needed to cause the immune response to generate extra thymic Tregs are still much debated. And I want to spend a couple of minutes talking about this. And about an interesting paper from Jim Allison that came out just a couple of months ago, published in the Journal of Experimental Medicine. So what he did was to take a TCR transgenic mouse, a CD4 line, and under in vivo conditions, he injected these TCR transgenic cells specific for moth cytochrome C, it's just basically a foreign antigen. And he had a number of peptides, synthetic peptides. This is the wild-type peptide, is strong, and he had two weaker variant peptides, which give up a poor immune response. So he just injected these peptides together with these TCR transgenic cells without adjuvant. It's a very important point. This is just peptide alone. And those are conditions that normally do not generate an efficient immune response. You know, you have to have an adjuvant to get an efficient immune response. So this is what we used to call signal 1 alone, where the cells will proliferate a little bit, then become anergic. Now what he did on the basis of typing cells for FOXP3 expression is he showed very dramatic production of Tregs in this particular situation. And so this is what he did. So he injects the T cells and then the peptide, and these are the peptides, and these are the weak peptides, and you see that if you looked at the proportion of the responding cells that expressed FOXP3 designating Tregs, weak peptides didn't do it, but this strong peptide, 60% of the cells after 2 weeks were Tregs. And this was not in the presence of TGF-beta or anything else, just a peptide. And this is what the data actually looks like if you use this CFSE. Everybody know what CFSE does? Okay, just a way of looking at division. And so these are the important data here. So you inject a peptide together with a low dose of this strong antigen, cytochrome C, and you're looking now at different days of proliferation, and you see that here at day 5 you've got massive proliferation going on here, and of the proliferating cells, a small but significant proportion, 9%, are Tregs. They express FOXP3. but the vast majority of cells don't. But under these conditions, where you don't have adjuvant, just peptide, these cells then die. And by day 15, they're almost all gone. But what you have left is these Tregs. And notice that a lot of these Tregs here have never even divided at all. They have not diluted out their CFSE. And so Treg production then is characterized by minimal proliferation, 1 or 2 or 3 divisions here. And these are the cells that resist undergoing actual death. And so after 2 weeks, most of the cells that you have are actually Tregs. Now this is with a low dose of this strong peptide. If you have a high dose of this strong peptide, you didn't see this. Then in fact the proportion of Tregs was much less. But with a high dose of this peptide, together with TGF-beta, which is an inhibitory molecule, which operationally makes a high dose of peptide into a low dose, then you see the same data as you see here. And that's probably the explanation for why TGF-beta is so good at generating Tregs. It basically reduces the intensity of the normal immune response. So as I said, this paper has only just come out, but I think it gives a new perspective of what are the requirements for generating Tregs during a normal immune response. Now what they didn't do in this paper, which they should have done, is to ask the question, well, what happens if you have an adjuvant, a TLR agonist? What do you see here in terms of Treg production? And they didn't do that. That'll probably be the next paper. Okay, so I'm going to finish off by considering what happens at the end of an immune response. Now, I'm not going to talk about memory. I'm going to leave that to other people to talk about. I want to talk about the end stages of a normal immune response. Now, typically, a normal immune response ends with the elimination of the pathogen concern. So if you're dealing with an acute infection, to say influenza, or in the mouse, lymphocytic choriomeningitis virus infection, you mount a very vigorous immune response, massive production of effector cells, the pathogen is destroyed, and then all these effector cells that you've generated in huge numbers are now not needed. And so if every time you got an acute infection, all the effector cells survived, you'd soon be full of the effector cells, which are basically in the way now. So evolution has determined that once these effector cells are no longer needed, after the pathogen has been eliminated, then they're destroyed. Why are they destroyed? Well, we don't know. Probably the simplest idea is that when the pathogen disappears, there's no more TCR stimulus anymore. And these effector cells really don't express many receptors for cytokines. So they can't receive a TCR signal, they can't respond to cytokines, and so they are neglected. And these neglected cells just die. And that's associated with upregulation or downregulation of BCL2 and upregulation of death molecules like VIM. So in an acute infection, 95% of the specific cells die, and just a small number of cells become resting memory cells. And I think Charlie Sir will probably talk about memory in later talks. Now, I want to talk about chronic infection because chronic infection is basically the continuation of an acute infection. So here the point to emphasize is that quite a lot of pathogens are not eliminated, unlike influenza and so on, and you actually have a chronic immune response. So some examples here: hepatitis, HIV, malaria, etc. And so you have a continuous response to these chronic antigens, and eventually the cells seem to give up, and they undergo what has loosely come to be termed cell exhaustion. And what's emerged in recent years is that exhausted T cells have been instructed to be unresponsive. And this reflects the fact that these cells which are anergic are now known to express a variety of cell surface molecules that are inhibitory. I mentioned CTLA-4 before, and these can be expressed at a high level on these exhausted cells. And then there are other molecules like PD-1, TIM3, BTLA, and so on. And there's a lot of interest in what these molecules actually do. And I've mentioned some of the functions of CTLA-4. And I want to finish off by talking about PD-1. And this has therapeutic implications because in diseases like hepatitis and malaria and so on, if you could overcome exhaustion, then you might be able to effect an efficient immune response and get a cure. But you have to think then about why do T cells express these inhibitory molecules? You'd think that it would be much better to not express these molecules so that the immune response would be efficient, and then you could eliminate these pathogens which normally get chronic infections. The problem here is that if the immune response to chronic infections is too vigorous, then you can have bystander damage and death from pathology. And that's illustrated by the 1919 flu pandemic. And that killed far more people than died in the First World War. There's some estimates that it killed in the order of 50 to 100 million people throughout the world. And the thing about this pandemic is that it killed mostly young, healthy people. Not the old and the young, but healthy people. And there's now good evidence that what happened during this pandemic is that it was such a powerful organism and which elicited such a powerful immune response that the T cells that responded, they would release all these pro-inflammatory cytokines, and then the patient would develop severe pulmonary edema and basically die from pulmonary edema, from drowning, in fact. And that's why it killed so many people. And you don't often see that these days. There's always a risk that through mutation we'll have another flu that mimics this. But let me just mention this peculiar episode that happened 2 or 3 years ago of this particular anti-CD28 antibody made by a company in Germany. Now it has this name, TGN-1412. And it was termed a super agonistic anti-CD28 antibody. And it was developed clinically to be used for stimulating Tregs, because in rats it was found that if you injected these antibodies into the rats, it would selectively expand Tregs. And so it was thought that this would be a useful treatment to treat autoimmune disease such as arthritis. And so when you do a clinical, if you want to use something clinically, you have to do clinical trials for toxicity, and it seemed to be fine in mice. And so what these people did, this company, is they asked for some volunteers, and they picked 6 volunteers, and they said, would you mind if we injected you with this apparently very safe drug, just to prove that it's not toxic. And these people almost died because there was a massive cytokine storm which was not seen in mice. And fortunately, people, they didn't die, they did recover, but this is what happened to one unfortunate person here. This because of the toxic shock. The blood vessels to his hands all closed down, and you see the gangrene here. So there must have been some powerful lawsuits that were filed, and in fact this company is now bankrupt. So that's one example where signal 2 alone can be enormously stimulatory. This doesn't happen, to my knowledge, with any other described anti-CD28 antibody. And why this one particular antibody has this dramatic effect, and apparently only in people and not in mice and monkeys, which is what it was tested in, I think is still unclear. Okay, coming back to PD-1. So the point to emphasize here is that one of the reasons that evolution has caused cells responding in chronic infections to upregulate inhibitory molecules is to prevent immunopathology. So in hepatitis, if you mounted an immune response that would really destroy the virus, you would destroy the liver and you would die. So PD-1. So how does it suppress T cell function? And here's what I'm just going to show you an example from Rafi Ahmed's group. So this is actually a photograph of one of his papers that I made, which didn't come out all that well. So broadly speaking, when a T cell sees a viral antigen presented by influenza, then signal 1 and signal 2 would lead to the generation of effector cells, production of cytokines, which will then kill the pathogen. The infection is resolved, and you'll generate long-lived memory cells. So this is a situation where you don't have chronic infection, and now these memory cells express high density of receptors for cytokines. CD122 for IL-15, CD127 for IL-7. Charlie Sir will talk about that, and these cytokines keep these cells alive. Now, this is chronic infection if you're dealing with certain strains of lymphocytic chorionic meningitis virus in mice, and then the cells become exhausted, and this is, at least in part, a reflection of what PD-1 is doing, interacting with a ligand on the APC. And what these guys have found is that if they add an antibody to PD-1, then this suppression is invigorated, and the cells can reacquire their cytotoxicity and eliminate the infection. And that's illustrated in this paper of Rafi Ahmed, published a couple of years ago now. And this is what happens in an acute infection for PD-1. You can see in grey that early on the cells will upregulate PD-1, and then when the virus is eliminated, PD-1 disappears. But if it's a chronic infection, shown in red, then it persists. But if they then inject it in mice, CFSE labeled cells, without an antibody that blocks PD-1 interaction, the cells will not divide. That's shown here, untreated. This is cells taken from the spleen, the liver, lung, and the gut. You see, most of the cells don't divide. They're exhausted. But if you inject an antibody to PD-1, you overcome that. The cells are reinvigorated, and they divide like crazy, and they will eliminate the pathogen. So I just want to finish off by talking about the issue, well, how does PD-1 function? Is it really an inhibitory molecule? Does it block cell signaling? And I just want to leave you with this interesting paper of Jeff Bluestone, published last year, where they found on the basis of two-photon microscopy that PD-1 acts in a very, very unusual fashion. So if you have normal CD8 cells, when they see antigen on the DC, they will move like this, and they will stop moving in order to form a synapse. And then they can mount an immune response. But what they found, in fact, is that if the cells have a high density PD-1, then these cells, they don't stop moving, and they just will zip past, and they can't form a synapse, and that's why they don't mount an immune response. So in that sense, they're not really reinvigorated. They just are basically blind to the infection. Okay, so I've gone a few minutes over, so I'm going to stop there. So I hope I've given you some sort of appreciation about what can and happen during interaction with APCs and what can go wrong. So, are there any questions that you'd like to ask me about?
So in mature state tissue, they always express the MHC class 1 and 2?
Yes.
Self-antigen or other antigen?
Well, they always express MHC. That's
with the
constituent.
peptide.
The peptide is what happens for a normal cell is that there's constant production and degradation of proteins, self-proteins in the cell. And when the proteins are destroyed, then these peptides tend to get loaded onto whatever MHC molecules are being generated within the cell. So all cells will express MHC molecules on the surface which are continuously plastered, covered with self-peptides. Now, it's not every molecule that's covered with a self-peptide. Some of the molecules are empty, and those are the ones that you can load with an exogenous peptide. But basically, all the molecules are filled with self-peptides.
So and then activate the costimulatory molecule and they give the signal to the T cell or B cell.
Yes. Yeah. Okay. Other questions?
Okay, thank you. Why don't we do the background?